Welcome to springlib.
Hi everyone, my name is Eric and let's answer problem 7. In problem 7, we are going to be focusing on the E. coli polymerase. Now, as you see that we are ending the word with ase, we can tell that the polymerase is an enzyme. Now, an enzyme, as it is going to carry out a function. And so a polymerase, its job is to carry out the function of transcription, where we are going to be converting DNA and building an RNA molecule. Okay? Essentially, transcription. The thing about the polymerase for the E. coli is that it is consisted of subunits. Subunits meaning smaller, different parts that can assemble together to make one polymerase. And specifically for the E. coli polymerase, it is made up of a 4-subunit combination where we have 2 different kinds of subunits coming together, but 4 different subunits in total. So let's go over the subunits and talk about their role in the polymerase and determine which subunit is going to be specific to transcription, meaning what gives the polymerase that specificity to the DNA in order to make transcription. So if this is our polymerase here, a 4-subunit polymerase, we can tell that we are going to have several different parts to it. And so over here, we are going to have what's called an alpha subunit. Okay. Now the alpha subunit is required for assembly, essentially putting together this polymerase. And you are going to have 2 alpha subunits inside of a polymerase. Additionally, we're going to have what's called a beta subunit. Now, the beta subunit is involved in the chain elongation, so extension. So when we are going to be building our RNA, this beta subunit is going to be responsible for extending out that new chain. Additionally, as one of the answer choices, we have a beta subunit prime. Now, this beta subunit allows for DNA binding, so connecting this polymerase to the actual DNA template. And finally, one of the final factors is that this subunit will come together with what's called a sigma factor. And the sigma factor is used to direct this enzyme to the promoter. So it brings this whole unit together to the promoter. And thus, this polymerase can bind onto our DNA, and then it will be able to transcribe and make RNA. Now, in order to answer the question, let's go back and review what they're asking for. We want to have something that allows for the polymerase to bind specifically only to the DNA. And the only answer choice that would be responsible for that is the beta prime subunit, because it allows for that specific DNA binding. So the answer to this question is going to be C, the beta prime subunit. Thanks for watching, and I hope this helps explain about the E. coli polymerase.